Alright, hello everybody. My name is Glenn Harter. I am the protocol expert here. Well, let's jump right into this. Like I said, protocol expert, so I'm doing the methods here. And I'm sure my partners stated that we have 5 different temperatures. So because of that, we obviously had 5 different copepod wells. So each well was set up in this sort of fashion, with rows A, B, C, and D, and we only used rows A and C, 1 copepod in each of the 6 wells, for a total of 12 in 1 well. Thus, a total of 60 for each trial, which is 5 times 12. There's a little diagram in the PowerPoint if you didn't understand that one. So that's basically how we set up the wells. All right, so before actually putting in the copepods in each well, we had to obviously get the copepods and then calculate the amount of algae needed for them, a week's worth of food. So we decided that the gender of the copepod didn't matter, and then we tried to get mature copepods, but we didn't really care if they were an algae either. And let's see here, in the case of the algae, the stock solution varied each week, so we had to recalculate the amount of algae needed using the hemocytometer. To show you what I mean, for example, using the hemocytometer in week 1, the concentration was found to be 760,000 cells per ml of algae per milliliter. And let's see here. You can see this little diagram here. We just kind of did a fraction here. We want this amount of food. This is what was calculated in the week 1. This is what was calculated to be in 1 milliliter of our stock concentration. So, since this is 1 milliliter, we can just divide this part. A little bit of mathematics, we come out to 0.46 mLs of our stock solution of algae. So that's basically how we did it. It was, pretty simple. So that's how we found the concentration and amount of algae we needed. So basically, after that was all said and done, we began setting up the wells. So we really didn't care which went first, the copepods or the algae. So that didn't matter. So yeah, we did that, and then the wells, each well only holds about 3 milliliters of solution. So what we did was, after we had filled the wells with the copepods and the algae, we filled the wells with 35 parts per thousand salt water to about 2.5, 2.8 milliliters of full capacity. Obviously, so it didn't overflow when we were carrying the wells over to the desk on the front, in the front of the classroom. So basically, after we did that and put the covers on the wells, we both double-checked that both food and copepod were in each well. Obviously, so we had our results for the next week. So that's basically it for the setup. The takedown of our experiment involved obviously taking the observations and data of what happened that week. With the surviving copepods, what we did was we put them back into the class solution of copepods, and then the dead ones we just rinsed out when we washed the wells. So basically improving the methods. Obviously, as we did it each week in and week out, we got better. But there were certain things that we did improve on. For example, obviously somebody was better at catching the copepods, somebody else was better at figuring out the concentration needed for each well, for the food, for the copepods. So we did that. We specialized our jobs. But in week 3, we discovered that 2 processes that made our methods quicker. So the live algae would move under the hemocytometer. So what we did was we nuked a sample of the algae to count it, so it would stay still under the hemocytometer, and so that increased the accuracy of the food needed. And then we also found out in week 3 that it was better to use the micropipette instead of the serological pipette. It just decreased the human error, and it was way more accurate, and it was also faster. So finally, our shortcomings I did mention that the gender or life stage really didn't matter for the experiment, but maybe Naupali are more sensitive to changes in temperature, so that could have affected it. Also, like I said, miscounting the algae before nuking it, all the cells would move, so it was kind of hard to count. That probably made it a little harder to find an accurate concentration for our food. And finally, before we use the micropipette, like I said, there was a lot of human error in using the serological pipette with a little crank on it. So basically, that's basically it for our methods. Thank you.